The universe is so vast it defies imagination, with trillions of stars shining across billions of galaxies, many of which are orbited by their own planets. The sheer scale of these numbers almost guarantees that we should not be alone. To answer this fundamental question, we must look back at the very beginning of life on Earth, tracing the long and improbable steps toward intelligence and what they imply for the odds of life elsewhere. Our journey follows the trail from the first chemical spark to the search for alien worlds and the mystery of the Fermi paradox. The story of life does not begin with organisms, plants, or even cells, but with chemistry. Before the first microbes appeared on Earth, there were only molecules moving in water. They collided, broke apart, and occasionally joined together, with most interactions being random and driven only by chance and environmental energy. However, in this primordial soup of reactions, something extraordinary happened. Certain molecules acquired the ability to create copies of themselves. That moment marked a turning point in history, the threshold where the long chain of evolution could finally begin. It is a long story, so let us start with the dream, and do not forget to like and share before we dive in. Chemistry usually follows blind rules, so how did it cross the line into something like biology? For life to exist, molecules must not only form but also preserve their patterns and pass them on. Replication is the secret that changed the game. Without it, the spark of complexity would be fleeting and vanish instantly. With replication comes continuity, and with continuity comes the opportunity for improvement. One of the leading ideas to explain this puzzle is the RNA world hypothesis. RNA is a close relative of DNA and plays multiple roles in living cells today. It carries instructions, helps build proteins, and can even fold into shapes that act like miniature tools. This makes it unusual because it can both store information and perform work. The theory suggests that before DNA and proteins took over, RNA molecules might have been at the center of the first living systems. They might have been able to replicate themselves, or at least parts of themselves. Thus, RNA may have been the foundation of life. Experiments have added weight to this idea. In the 1980s, scientists discovered that certain RNA molecules called ribozymes could act as catalysts. They were no longer passive carriers of information but could speed up reactions just like proteins do today. This was unexpected because it was long believed that only proteins possessed such capabilities. If RNA could both record its own sequence and assist in assembling copies, it seems a natural candidate for the earliest replicator. But the RNA world is not without its problems. A major issue is how RNA molecules were synthesized in the first place. RNA is made of smaller parts called nucleotides, and forming these under prebiotic conditions is not simple. Even if they could appear, linking them into long chains is another challenge. In the lab, scientists have induced RNA molecules to form and even replicate fragments, but a fully self-sustaining cycle remains elusive. Critics argue that RNA might only explain part of the story and may not have been the starting point. One view suggests that simpler self-replicating molecules existed before RNA. Some researchers propose that short peptides might have played this role. Unlike RNA, peptides form more easily under early Earth conditions, especially near volcanic vents or in pools that repeatedly dry out. If these molecules could arrange themselves into repeating patterns and promote their own reproduction, they might have provided the stepping stones for more complex systems like RNA and DNA. Another possibility is that the first replicating system was not a single molecule at all but a network of chemical reactions. Instead of one RNA chain carrying everything, it was a collective of molecules assisting each other in self-replication. This is known as the metabolism first idea. From this perspective, self-replication is not a single event but an emergent property of a chemical network. If certain reactions produce molecules that fuel other reactions and form a cycle, the system as a whole can maintain itself. Over time, these networks might become more stable and eventually give rise to informational molecules like RNA. On early Earth, minerals like clay could provide structured surfaces for molecules to align. If certain molecular patterns adhered to a surface and new molecules attached in the same order, this could be an early form of copying. The mineral itself does not replicate, but it acts as a template, helping chemistry move toward a more organized direction. Replication is not just about making a copy, it is about making a copy that preserves enough information to keep the system stable, while allowing for tiny changes that open the door to evolution. This process of applying pressure for improvement is the very beginning of Darwinian evolution, long before cells or genes appeared. The mystery remains unsolved because evidence is scarce. Traces of those first molecules vanished long ago, leaving behind only indirect clues in modern life and laboratory results. Nevertheless, progress continues. Chemists have designed synthetic molecules in test tubes that can self-replicate. These artificial systems are much simpler than RNA or proteins, yet they prove that self-replication is not a miracle but a process that can emerge from chemistry under the right conditions. Perhaps molecules did not leap into perfect replication but stumbled across small steps, a fragment that occasionally helped reassemble itself, a surface that favored repeating patterns, or a network of mutually reinforcing reactions. Each link added a layer of persistence until one day a molecule or a group of molecules crossed the threshold and became what we can truly call alive. If replication is a natural consequence of chemistry under the right conditions, then life might not be rare. On the other hand, if reaching the stage of replication requires a series of highly improbable events, then life may be more precious and fragile than we realize. As the mystery of replication began to clear, another obstacle stood in the way of life. Even if molecules could copy themselves, they were still exposed to the raw forces of the environment. The early Earth was not gentle. Waves, heat, lightning, and radiation constantly destroyed fragile structures. Molecules floating freely in the ocean struggled to stay together long enough to become more complex. To survive, they needed a sanctuary, and this is where the story of the membrane begins. The rise of tiny protective bubbles might have been just as important as replication. A membrane is a boundary. Today, every living cell has a membrane, primarily composed of lipid molecules. The special thing about lipids is their dual nature: one end is attracted to water, and the other repels it. When many lipids are placed in water, they spontaneously arrange themselves with the water attracting ends facing out and the water repelling ends facing in. This spontaneous behavior closes into a bubble, forming a vesicle, a soft, tiny pocket that can trap molecules inside while keeping the outside world out. This requires no designer, it is simply the natural behavior of lipids under the right conditions. Moving from free-floating molecules to being sheltered by bubbles was a new stage on the path to life. Inside these vesicles, chemistry could proceed in relative safety. Instead of being washed away by waves or destroyed by external energy, molecules inside the vesicle had a chance to interact. These compartments provided stability and identity. A bubble containing a unique combination of molecules could now be seen as a single unit. Experiments show that under simple conditions, lipid bubbles form very easily. When scientists mix fatty acids or similar molecules with water, vesicles appear almost automatically. Fatty acids on the early Earth could have come from many sources, such as reactions near hydrothermal vents or even chemical delivery from meteorites. Some studies have found lipid-like compounds in meteorites, suggesting the raw materials for membranes might have come from space. Whether synthesized on Earth or arriving from elsewhere, these lipid molecules possess a stunning property: self-assembly. They can build compartments without any command. The importance of this cannot be overstated. For life to advance, molecules needed to be both protected and concentrated. A thin chemical soup dispersed in the ocean would never reach the complexity of life. Vesicles solved the problem of dilution by gathering molecules in one place. Reactions that were rare in the open sea became more frequent in small spaces. A bubble might capture RNA fragments, amino acids, and minerals all at once, providing a stage for them to interact. The membrane thus became a tool for gathering ingredients. Another striking feature of these primitive membranes is that they can grow and divide. When more lipids are present in the environment, fatty acid vesicles expand. If they are shaken or stretched, they can split into two smaller bubbles, sharing the original contents. This simple physical process foreshadows what cells do today when they divide. In the beginning, no complex machinery was needed. The physics of the membrane itself allowed for a primitive form of reproduction. Imagine a vesicle that happens to trap an RNA molecule with catalytic abilities. Inside the bubble, this RNA could help produce more of itself or help synthesize useful molecules. When the vesicle divides, the new bubbles carry copies of the RNA with them. Over time, the physical and chemical combinations that were more efficient at this would naturally become more common. This coupling of molecules and containers was a key step from scattered chemistry toward organized life. This view is supported by ongoing research into protocells, which are laboratory models of the first life compartments. Scientists have built simple vesicles that can trap RNA, allow small nutrients to pass through, and even undergo cycles of growth and division. These protocells are not fully alive, but they give us a glimpse of how life became more than the sum of its parts once it was contained. There is still debate about the composition of the first membranes. Modern cells use complex phospholipids, which might have been hard to form on early Earth. Simpler fatty acids are more likely candidates. They form vesicles under a wider range of conditions but are also more fragile. High salt or extreme heat can break them apart. This suggests that early life might have been limited to environments where membranes could survive, such as freshwater ponds or protected shorelines rather than the open ocean. The choice of habitat was not a preference but a matter of chemistry. For life to continue, molecules also had to go in and out. A perfectly sealed bubble would trap its contents forever, while one too loose would fall apart. Fatty acid vesicles seem to strike a balance, allowing small molecules like nucleotides and ions to pass through while keeping larger molecules inside. This permeability might have made early protocells unexpectedly active, as nutrients could flow in and waste could flow out, allowing the internal chemistry to evolve. Some researchers also propose that the membrane surface itself served as a reaction site. Molecules adhering to lipids might be held in specific orientations, making reactions easier. In this way, the membrane was not just a container but a participant in early chemistry. As protocells became more complex, the partnership between the membrane and replicating molecules likely grew tighter. RNA fragments that could stabilize or promote membrane growth would be favored, and vesicles carrying beneficial RNA would be more likely to survive and divide. This coevolution of container and content likely drove the first steps toward true cells. The chaos of early Earth was not eliminated but channeled into countless tiny safety zones, allowing life to experiment with itself. Among countless fleeting and fragile bubbles, a few carried the seeds of persistence. To this day, every living cell carries the memory of this breakthrough. Our own membranes, with their complex proteins and channels, are the direct descendants of those first simple bubbles that learned to maintain the gradients necessary for life. Without a stable flow of energy, even the most promising protocell would be a temporary structure that collapses once its initial resources are exhausted. Life, from the very beginning, had to learn to capture energy and convert it into useful work. The forms of energy on the young Earth were diverse. Sunlight poured down from above, though the atmosphere was different then, allowing more ultraviolet light to penetrate. In the deep ocean, volcanic activity churned with heat and chemicals, constantly changing the underwater environment. Thunderstorms split molecules in the air, creating new ones. These were the primitive power sources. Life's task was to turn these uncontrollable forces into structured ways of using energy. The bridge between chaos and order is the gradient, the difference in energy from one place to another. A gradient means energy is not distributed evenly, and any system lucky or clever enough can exploit it. One of the most compelling scenarios occurs near hydrothermal vents on the ocean floor. These vents, driven by volcanic heat, spew hot water rich in minerals and chemicals, creating sharp temperature and chemical gradients. Hot water meets cold seawater, and acidic fluids meet alkaline fluids. The vent walls also contain minerals that can act as natural catalysts. In such environments, protocells might find both shelter and energy. Alkaline hydrothermal vents, in particular, are highlighted as natural laboratories for the origin of life. Unlike the black smokers that release scorching acidic fluids, these release cooler alkaline water. The chimneys formed around them are full of tiny mineral pores that act as natural compartments, much like primitive membranes. More importantly, they establish sharp chemical gradients, with high alkalinity inside the pores and more acidic seawater outside. Across these thin walls, a natural difference in proton concentration exists. Modern life still relies on proton gradients to function. Every living cell, from bacteria to humans, uses membranes to pump protons to one side and then uses that cross-membrane concentration difference to generate ATP, the energy currency of life. The fact that life still uses this system today suggests its roots are extremely deep. In the vents, the gradient existed naturally, and primitive chemistry might have utilized it long before cells evolved their own proton pumps. The micropores of the vents acted like ready-made batteries providing low-level usable energy. Before the rise of atmospheric oxygen, the ultraviolet component of sunlight was much stronger. To us, this seems like a destroyer, but it might have been useful. Ultraviolet light is strong enough to break chemical bonds, driving reactions that otherwise would not occur. Experiments show that UV light can help form nucleotides, amino acids, and other building blocks of life. It could both destroy fragile molecules and assemble new ones. If protocells found ways to shield or filter the light, they might have used this raw energy for themselves. Different protocells likely explored different energy strategies. Some stayed near vents, utilizing proton gradients and minerals, while others relied on sunlight for light-driven reactions near the surface. Over time, more stable or efficient energy sources were favored. The competition for energy was likely the first great selection pressure. To grow larger, replicate information more reliably, or form stronger membranes, protocells needed more energy than diffusion alone could provide. Once a system found a way to consistently channel energy, it could outcompete its neighbors. The emergence of energy utilization marked the boundary between accidental chemistry and true ancestry. In today's cells, energy is often obtained by moving electrons. For example, in respiration, electrons are passed along a protein chain, releasing energy step by step. Minerals like iron and sulfur on the early Earth might have provided the original versions of these chains. Electrons flowing across mineral surfaces, coupled with natural gradients, might have driven the earliest biochemical cycles. This deep connection between geology and biology suggests that life's first engines were not invented out of thin air but were borrowed directly from the planet. Earth already had systems for moving energy through vents, gradients, sunlight, and mineral reactions. Life's task was to capture these energy flows and trap them within a membrane. Once trapped, energy could be used in an orderly way to link nucleotides into RNA, amino acids into peptides, or to reinforce the lipid membrane itself. Energy turned fleeting chemistry into a lasting process. Under the icy moons like Europa or Enceladus, hydrothermal vents might exist beneath the ice crust. If gradients are the key, similar processes might be happening today, perhaps giving birth to life or sustaining what already exists. On planets closer to stars, light-driven chemistry likely plays a role. As long as there is an energy source, chemistry, and a compartment, the recipe for life is not far away. The energy in the early stages was often chaotic and even destructive, but even a protocell that could slightly channel it would gain an advantage. After countless generations, these edge cases became the norm, and simple cycles grew into complex ones. The initial reliance on natural gradients eventually evolved into the sophisticated energy systems we see today. For hundreds of millions of years after life appeared, this planet was a completely different world. The oceans were full of simple cells living in a world without oxygen. To them, oxygen was not the breath of life but a poison. The atmosphere then was mainly composed of methane, carbon dioxide, and nitrogen. Early life found ways to survive through chemical reactions that did not involve oxygen. It was a world of anaerobic organisms, maintaining a long-term balance. Then, something dangerous and extraordinary happened. A new type of microbe learned to use sunlight in a different way. These microbes were the ancestors of what we now call cyanobacteria. In the process of photosynthesis, they learned to use water as a source of electrons. This was a massive leap in evolution. By stripping electrons from water molecules, they could fuel their own growth and release oxygen as a byproduct. To them, it was merely a waste gas. The oxygen released by these early photosynthesizers was initially absorbed by iron and other substances in the ocean. The iron was oxidized, forming the banded iron formations we still see in ancient rocks today. For millions of years, the oceans were quietly filling with oxygen, but it remained locked away by these chemical reactions. Eventually, the absorption capacity was overwhelmed. The iron was exhausted, and oxygen began to leak into the broader environment, slowly changing the oceans and the atmosphere. For the organisms that had thrived in the old world, this was a catastrophe. Oxygen, a highly reactive gas, could tear through the delicate chemical bonds inside cells, destroying proteins, nucleic acids, and membranes. To these anaerobic beings, the rising oxygen levels were lethal. A once thriving biosphere was suddenly exposed to a gas it could not tolerate, turning lush habitats into killing fields. This event, known as the Great Oxidation Event, nearly ended life before it could truly expand. The scale of this crisis is hard to imagine. It was not a sudden disaster like an asteroid impact, but a chronic poisoning that lasted millions of years. Wherever oxygen reached, anaerobic life either died out or retreated to hidden sanctuaries where oxygen could not penetrate, deep silt, underground cavities, and low spots on the ocean floor. Some believe that during this period, Earth lost most of its biosphere, as if the planet itself had suddenly turned hostile to the life it had nurtured. However, crisis also brought opportunity. Although dangerous, oxygen carried immense energy. Chemical reactions using oxygen release far more energy than anaerobic ones. Those organisms that could endure or even utilize oxygen suddenly possessed entirely new possibilities. Evolution is always a game of survival and adaptation, and life began to explore this new path. The first step was defense. Cells developed enzymes and molecules to neutralize oxygen toxicity, creating primitive antioxidant shields to protect their inner machinery. Over time, these shields grew stronger, and some organisms did more than just survive, they learned to harness the gas. The shift from anaerobic to aerobic metabolism is one of the most profound changes in the history of life. With oxygen, cells could extract far more energy from the same amount of food. This extra energy opened the door to complexity. The rise of aerobic life did not happen overnight, it was a process of trial and error and countless failures. But gradually, the lineages that mastered oxygen surpassed those that could not. This new metabolism allowed cells to grow larger, move faster, and build more intricate structures, setting the stage for multicellular life, even if its arrival was still far in the future. The Great Oxidation Event also drastically affected the planet itself. With oxygen in the atmosphere, new chemical cycles began. Methane, once a crucial greenhouse gas, was destroyed by oxygen, causing the Earth to cool. Some believe this triggered the first Great Ice Age, freezing the planet from pole to pole in what is called Snowball Earth. If so, oxygen reshaped the environment with even more challenges. Life had to endure poison, ice, and darkness all at once. Despite this upheaval, the seeds of the modern biosphere were sown. Oxygen eventually became a permanent fixture in the atmosphere, and new ways of breathing became the standard. An entire ecosystem powered by oxygen emerged. Over time, the gas that once threatened to destroy life became the primary driver of its expansion. The gas we depend on so completely today was once the culprit of a radical extinction. The survivors moved forward with the tools to coexist with oxygen, and their descendants built the complex world we see now. Every breath we take is a reminder of that ancient disaster and the resilience of early life. Progress often comes at a cost, and new inventions can be fatal to the existing order. The cyanobacteria did not intend to poison the world, they were simply solving the problem of their own survival, yet they triggered a global transformation that forced everything else to change or perish. For billions of years after the origin of life, Earth was dominated by simple cells. These prokaryotes were small, efficient, and resilient, existing everywhere from the deep sea to rock surfaces. But despite their success, they remained simple, limited in structure, size, and energy budget. Then, about 1.5 billion years ago, a simple cell swallowed another. But this time, it was not digestion, it was a partnership. This union gave birth to eukaryotes, cells with a nucleus and compartments that could grow to unprecedented sizes. It is not unusual for cells to eat each other, but what was extraordinary here was that the swallowed cell did not die. Instead, it found a way to survive inside the host, and the host benefited from its presence. The swallowed cell is what we now know as the mitochondria, the miniature power stations inside every complex cell. From that point on, the energy supply was permanently changed. Mitochondria are remarkable because they convert oxygen and food molecules into far more energy than a simple cell could produce on its own. This energy leap allowed eukaryotes to grow larger, build more structures, and carry more DNA, much like switching from a candle to a power generator. With this surplus of energy, cells could afford complexity. They built protein skeletons, transport systems, and specialized compartments. The nucleus appeared, providing a safe home for DNA and regulating its use. Other compartments evolved, but the core remained the mitochondria. Without this endosymbiosis, there would likely be no multicellular organisms, plants, animals, or humans. What is even more shocking is that this event seems to have happened only once. Despite billions of years and countless opportunities, there is no evidence that a similar union ever occurred in other lineages. Prokaryotes continued their success, but only one lineage made the jump to true complexity by taming another cell. Why is it so rare? One reason might be the incredibly delicate balance required for such cooperation. Consumption usually ends in destruction. For the swallowed cell to survive, the host must tolerate the invader, and the invader must adapt to living inside another. Even if this balance is achieved by chance, it must persist across generations. Both parties must reproduce in sync. Its success is nearly a miracle. Mitochondria still hold clues to this past. They carry their own set of free-floating genes independent of the cell nucleus. Though this DNA is small and simplified, it reveals that mitochondria were once free-living bacteria. Over time, most of their genes were lost or migrated to the host nucleus, leaving only a few behind. They became completely dependent on the host, unable to survive alone, and the host in turn became dependent on their function. The two became one. With this energy dividend, eukaryotes expanded into forms that prokaryotes never touched. Some single-celled individuals grew as large as grains of sand, while others began to work together to form the first multicellular organisms. More energy meant more DNA, more proteins, more regulation, and more innovation. Algae, fungi, plants, and animals all came from this. All the complexity we see can be traced back to that improbable union. This leads some scientists to wonder if eukaryotes are a fluke of our planet. If life exists elsewhere, it might stay as simple cells forever. If that step is a one-time occurrence, then the prevalence of intelligent life might be far lower than we think. Yet, this leap also carries a lesson of cooperation. We often see evolution as competition and survival of the fittest, but the birth of eukaryotes is a story of partnership. We are, at the deepest level, symbiotic beings. In plants and algae, symbiosis happened again later. A eukaryotic cell swallowed a photosynthetic bacterium, which became the chloroplast. Like mitochondria, chloroplasts keep their own DNA. This second great union created the plant kingdom. But the first round with mitochondria was the foundation. Without that energy burst, it would have been impossible to support another partner. These layered events highlight how narrow the path to complex life truly is. Along with the nucleus and larger genomes, eukaryotes developed sexual reproduction, mixing genes faster and accelerating diversity. This allowed for rapid adaptation and eventually led to the explosion of forms in the fossil record. Looking back is humbling. All animals, plants, and fungi share this ancestry. The rise of eukaryotes was not inevitable, it was a fragile, one-off event. If we look at Earth's timeline, we must realize how much of it belonged to microbes. Earth is about 4.5 billion years old, and for more than 3 billion of those years, the planet was occupied only by bacteria and other simple organisms. Large plants, animals, and even multicellular life appeared only at the very end of the story. If you compress Earth's history into a single calendar year, then from January to late November, the planet was inhabited almost exclusively by single-celled microbes. Everything else, forests, dinosaurs, mammals, and humans, only appears in the final month or even the final week. The first eukaryotic cells might have opened the door to getting bigger, but the jump from a single complex cell to a multicellular organism did not happen immediately. Instead, eukaryotes drifted in the ocean for over 1 billion years without producing the structured complexity that led to animals and plants. Scientists sometimes call this period the boring billion. It was as if biology was waiting for something. Perhaps the environment wasn't ready. Complex life needs a steady supply of oxygen, not just in the atmosphere but dissolved in the ocean. If oxygen levels were too low, large, high-energy life forms could not survive. Evidence suggests oxygen rose in steps rather than all at once, with long pauses in between. If this is true, evolution was just waiting for the right conditions, like wood that is too damp to catch fire. Another explanation looks at biology itself. Moving to multicellularity is not a small step. It requires cells to give up their independence and cooperate as part of a larger whole. This means some cells must specialize and stop reproducing to serve the collective. From an evolutionary standpoint, this is a massive sacrifice. Microbes are excellent survivors on their own, they don't necessarily need complexity to succeed. Perhaps in that long gap, life tried to become complex many times, but the evidence was erased. Multicellular experiments may have appeared and vanished, ground down by environmental shifts or competition, until finally one experiment succeeded and spread globally. If Earth's story is representative, the universe might be full of microbial planets that never take the step toward complexity. Then, about 540 million years ago, Earth underwent an extraordinary transformation. After billions of years of life being nearly invisible, the oceans suddenly exploded with diversity. Creatures with shells, spines, and eyes appeared abruptly. This was the Cambrian explosion. A burst of creativity filled the sea with amazing forms. Sites like the Burgess Shale in Canada and the Chengjiang fossil site in China reveal a world of experimentation, strange spiked worm-like creatures, odd arthropods with grasping appendages, and the earliest ancestors of mollusks and vertebrates. Why so sudden? One trigger might have been oxygen finally crossing the threshold needed for high metabolism. Active animals with muscles and nervous systems need the spark that oxygen provides. Once oxygen diffused into the oceans, it may have unlocked a biological arms race. Another explanation focuses on the genetic toolkit, the set of genes that governs how a body is built. These genes determine where limbs grow and how organs are laid out. Remarkably, these gene families are found in everything from flies to humans. Before the explosion, this toolkit might have been simple or underutilized. During this period, it may have expanded or changed, allowing for greater flexibility in body design. With new instructions, organisms could evolve eyes and shells more rapidly. At the same time, shifting continents and rising sea levels created perfect laboratories for evolution in shallow seas. Geologic evidence shows ocean chemistry changed as well, releasing nutrients like calcium that allowed for the formation of hard shells and skeletons. Once some creatures had protection, others had to respond by developing ways to drill through shells or move faster. Complexity bred complexity. The appearance of the first true predators changed the rules of survival. If someone is hunting you, you must defend yourself, hide in the mud, or grow a shell. This arms race was a key driver of rapid innovation. While the Cambrian explosion was dramatic, it wasn't instantaneous. Roots of this complexity were already stirring in the earlier Ediacaran period, with strange, soft-bodied forms. The Cambrian was the moment these experiments finally solidified. If complexity requires such a specific convergence of oxygen, genetic innovation, and environmental change, then complex life might be rare in the universe. But on the other hand, it might show that once conditions are met, complexity is almost inevitable. Life fills every ecological niche as soon as it has the energy and space to do so. Yet, in this long history, intelligence was never guaranteed. For billions of years, organisms survived and thrived without high-level thought, relying on simple nervous systems and basic senses. Sharp vision, strong muscles, hard shells, or a keen sense of smell were often the only differences between life and death. However, within one small lineage of primates, something shifted. Thoughts became deeper, and awareness became more complex. Eventually, the capacity for reflection, planning, and imagination transformed survival strategies into something far more profound. Dinosaurs ruled the planet for over 150 million years, yet they never developed writing, agriculture, or technology. Insects became the most numerous and successful animals on Earth, but their survival depends on instinct-driven behaviors. Even mammals that evolved complex brains mostly remained at a level of consciousness suited to their specific ecological niches. Wolves can hunt in coordination, dolphins communicate with clicks and whistles, and elephants remember ancient migration routes. Yet, none of them truly advanced toward symbolic thought, abstract language, or deliberately developed culture. 4. Most of the history of life, being smart was not the primary currency of survival. Speed, strength, and resilience mattered more. This is why the rise of human intelligence is both extraordinary and puzzling. One view is that intelligence is just one natural selection among many, like a branching tree where different traits work in different contexts. Birds learned to fly, fish evolved gills, and plants invented photosynthesis. Each path is viable, with no absolute hierarchy between them. For humans, intelligence might just be another adaptation that happened to work in the specific environment our ancestors faced. Early primates lived in forests where three-dimensional vision, root memory, and social awareness were highly useful. As some groups left the trees to adapt to life on the savannas, new challenges appeared. Food was more scattered, and predators had the advantage in open terrain. Cooperation became vital. In such an environment, sharper thinking might have been a self-reinforcing advantage. The role of social living is often emphasized. Many animals live in groups, but primates pushed this complexity to an unusual degree. To survive in a group, one must constantly remember allies and rivals within an ever-shifting hierarchy. A brain that could interpret subtle gestures, predict behavior, and manipulate social connections gained an advantage. This idea is sometimes called the social brain hypothesis. It suggests that intelligence did not initially grow to solve problems in the environment, but to solve problems within the group. Over time, these mental tools were turned outward and applied to hunting strategies, tool use, and eventually, symbolic language. Tools themselves were likely another driving force. Millions of years ago, early hominids began shaping stones into cutting edges. Initially, these tools were simple and crude, but they extended the strength of the body, allowing us to access new food sources like bone marrow and fish. Making tools requires foresight, memory, and imitation. Teaching the use of tools, in turn, requires communication and instruction. A feedback loop was born. Smarter individuals made better tools, and better tools rewarded smarter individuals. Over countless generations, this loop likely pushed the brain to a degree that species not reliant on external objects could never reach. However, intelligence comes with a cost. The brain is massive and consumes an incredible amount of energy. In humans, the brain uses about 20% of the body's total energy at rest, which is much higher than in most animals. For this heavy burden to be worth it, the benefits had to outweigh the costs. Our ancestors found themselves in an environment where flexible thinking repeatedly paid off, whether in finding food, organizing hunts, or building alliances. But such an environment might not be common elsewhere. If conditions were only slightly different, if food were more abundant, predators fewer, or cooperation less necessary, the path to higher thought might never have been taken. Is human intelligence inevitable or accidental? Some argue that given enough time, evolution tends toward complexity, implying that intelligence will eventually appear on many worlds. Others warn that intelligence is a rare product of a chain of unlikely events. The asteroid impact that wiped out the dinosaurs 66 million years ago was one such event. Without it, mammals might have remained small and marginal, never expanding to experiment with new life forms. Specific climate changes that shaped the African landscape, ecosystem pressures, and the unique biology of primates might have all combined in just the right way. Even within our own lineage, the story is fragile. Multiple species of the genus Homo once existed simultaneously, including Homo habilis, Homo erectus, and Neanderthals. They all showed signs of intelligence, tools, and social life, but only Homo sapiens pushed development further, building symbolic art, organized language, and long-term culture. Why? The debate continues. Some believe a tiny genetic change made our brains exceptionally flexible, others emphasize cultural accumulation, where each generation adds a little to the previous one, creating a snowball effect. If so, intelligence was not a sudden leap but the result of ideas layered upon ideas over hundreds of thousands of years. Looking across the animal kingdom, we see glimpses of this potential. Crows can shape sticks into hooks to fetch food, octopuses can solve puzzles and escape containers, dolphins learn tricks and pass them to their peers. These sparks show that intelligence is not unique to humans, but in other creatures, it remained constrained. Their environments and lifestyles did not demand more. In contrast, humans were pushed into a narrow corner where only flexible thinking, social cooperation, and long-term planning could open a path to survival. From this narrow road, language, art, and technology emerged in a symphony unmatched by any other species. The greater mystery remains: is our intelligence a universal destination of evolution or a fragile rarity? If it is universal, the galaxy may be full of civilizations. If it is rare, we might be alone, a strange accident on a small planet. What is certain is that intelligence is not an easy path. For billions of years, life did not need it, and even today, most species thrive without ever building symbols or tools. The road from living being to thinker is narrow, winding, and full of uncertainty. That it happened at all is one of the most stunning turns in the history of life. That is all for today's knowledge sharing. I wish you all sweet dreams and a wonderful day ahead. We will see you in the next episode.